Okay, we'll get started then. Welcome. My name is Yanina Aldao. I am an incoming transfer student, and I am currently part of a team working at Dr. Nagel Lab in the Department of Botany and Plant Science at UCR. My team members are George Echeverria and Melvin Hodanu, right over here, and we are studying clock-regulated genes and their response to heat stress in rice. Rice is one of the fundamental pillars of human nutrition, yet climate change with its extreme heat waves, drought, and flood disasters is exacerbating the already strained rice production. In order to adapt to a changing climate, we need to understand the biological processes, pathways, and mechanisms underlying plant growth and development and its interaction with the environment. Our research this summer aims to understand how plants' internal clock regulates some heat stress responsive genes in rice. We all know that organisms are exposed to changes in the environment. One of the mechanisms used by plants to respond to environmental stresses is coordinated through an elaborate gene regulatory network that involves the circadian clock. You can see in this diagram here that the circadian clock controls many important plant processes, such as photosynthesis. You can see in this video how the sunflower is trying to maximize photosynthesis by following the sun through its journey in the sky. Other important processes controlled by the circadian clock are growth and blooming periods. So here you can see an overview of our 10 weeks research. For heat stress responses, understanding how time of day and the circadian clock alters transcriptional responses, such as the occurrence and magnitude of a response, is essential. For our summer project, we used an assay to characterize 3 responsive genes. The first approach on panel A was a reported based assay, which involved cloning gene promoters, fusing them to luciferase, and then infiltrating tobacco leaves to monitor changes in expression. The second approach on panel B was actually a backup plan that we had to use because of our luciferase camera being broken. And this actually gave us the opportunity to have a double check. We were able to look at gene expression using quantitative real-time PCR after heat stressing rice seedlings at 2 different times of the day. We will go further into detail on the next few slides.
Before entering the lab, we were each provided with a unique gene by Dr. Nagel. The genes that were selected were based on previously documented experiments in Arabidopsis looking at heat stress responses during different times of the day. Each gene we chose is either involved in thermal tolerance, as we saw in HSFA7A, or in growth and development, as we can see in CDF1 and TRFL3. The graphs on the screen right now are looking at RNA sequence analysis, showing the expression of these genes in response to heat stress throughout the day. As you can see, every single one of the genes that we were provided was significantly upregulated based on that previous data. Our research objective for the summer was to determine whether copies of these genes are also responsive in heat stress or to heat stress in rice plants. To begin our first experiment, we identified copies of the rice gene that we were going to select based on the Arabidopsis gene we were provided. Using these identified genes, we created and ordered primers that were complementary to our gene. These primers were then used to perform a PCR, and the product was ran through a gel using a method known as gel electrophoresis. In our gel, we placed our PCR product as well as a DNA ladder, a positive control, and negative control. If our reaction was successful based on the gel electrophoresis, we then cut out the band and purified our product. Using that purified product, we cloned it into our plasmid, P-enter D, and then plated it on LB containing kanamycin. A colony PCR was then used to verify the plasmid, and finally, we used sequencing to confirm the cloning of our rice gene.
Here you can see, this was one of our main assays. It's a high throughput process, which is an approach that can rapidly identify genes that modulate a particular biomolecular pathway instantaneously. We used a luciferase-based reporter assay for monitoring circadian rhythms over multiple days in plants. The idea here is that you could take a gene that you know is regulated by the circadian clock and fuse this to a reported gene such as luciferase. Luciferase are naturally occurring protein enzymes from the firefly that can catalyze emission of light from substrate. In this case, we transform the rice promoters, fuse them to luciferase, and transform them into agrobacterium, and then infiltrated tobacco leaves with this culture. This allowed us to monitor the activity of the gene promoters over time based on changes in brightness of luminescence. As you can see, we were only able to see the expression for a short period of time, about 6 hours, if you can see in the linear graph at the bottom. And this is because the promoter constructs were likely silenced by the tobacco plant. So we plan to repeat this experiment.
Next, we looked at the expression of these rice genes after heat stress. We subjected 2-week-old seedlings to a heat stress at 2 times in the day, at 9 a.m. and at 3 p.m. The graph on the right is just to show these corresponding times. Based on the analysis, we observed that the genes show specific responses to heat stress with some indication of time of day control. In addition, we were also able to do an in silico analysis to gain more insights into our genes. We also looked at published cold stress data, and we observed that our genes are also misregulated in response to cold stress. In conclusion, we found out that HSFA2C is always upregulated in response to heat stress and downregulated in response to cold stress. DOF12 appears to respond differently to heat stress in the AM versus PM, as compared to Arabidopsis, where it is upregulated throughout the day. TRFL3-like shows opposite response to heat stress in Arabidopsis and rice, and is downregulated in response to cold stress. To determine whether the rice plants were just expressing these genes, or they are more susceptible or resistant to heat stress depending on type of day, we plan to look at this further in the future by using techniques like survival assays. We would like to thank NIGO lab members, specifically Sabrina Gilmore, Samantha Crowley, Samantha Cordinley, Emily J. Blair, and Titwan Bonnet. And of course, our principal investigator and mentor, Dr. Dawn, for all of her support and sharing her knowledge with us. We also like to thank the Bailey Cyrus Lab, RISE coordinators, and peer mentors. And we give a special thanks to the NSF and PERSIST programs for funding our research.